Assalamu alaikum and good morning everyone. My name is Muhammad Umar and today our discussion topic is glycolysis. Dear students, a lot of chemical reaction take place in the body of living organism. Glycolysis is one of the important chemical reaction which take place in our body. Students, we take different kind of food like carbohydrates, protein and lipids, and the purpose of taking this kind of food to get energy. So in today's lesson we will discuss how body get energy from carbohydrates. Actually, when we take carbohydrates, body break down the complex molecule of carbohydrate into a simpler one unit which is called glucose. And glycolysis is a process in which a glucose molecule break and energy will be released in the form of ATP and NADH. So let's start to our lesson. So student, let's about our lesson which is about glycolysis. This is my channel Study with Umar. Please subscribe. The link will be given in the last slide. So there are some objectives of today's lessons. First one is the definition of biochemistry, metabolism, anabolism and catabolism. Second is to understand how the glycolytic pathway is used to convert glucose to pyruvate. The third is to understand the conversion of chemical potential energy in the form of ATP and NADH. And in the last we will discuss about the intermediates, enzymes and cofactors of glycolytic pathway. There are a lot of enzymes involved in glycolytic pathway, so all these enzymes will be discussed in this lesson. Definitions. First definition is the biochemistry. Biochemistry is a branch of science in which we deal with the chemical process within and related to the living organisms. Biochemistry is a combination of two words, bio and chemistry. Bio means life and chemistry means chemical reaction, means all the chemical reaction which take place in the living organism will be studied in biochemistry. Second definition is a metabolism. The sum of chemical reactions which take place in living organism is called metabolism. Of reactions are two types. First is a anabolism and second is a catabolism. Anabolism means small molecules are combined into a large one. Energy is required for anabolisms. Actually, anabolism is a joining process. In this procedure, small molecule combine and large or big molecule will be formed. The simplest example of anabolism is photosynthesis. In photosynthesis, carbon dioxide and water will be joined in the presence of chlorophyll and sunlight, and they produce glucose, which is a big molecule. Second is a catabolism. In catabolism, large molecules are broken down into a small ones. Energy is released, and the example of catabolism is respiration. Now, what is glycolysis? Glycolysis is a set of reactions that convert glucose to pyruvate or lactate. Glycolysis is a Greek word. Glyx means sweet and lysis means splitting. So glycolysis is a splitting of glucose. There is an equation of glycolysis. Glucose molecule will be break down into 2 pyruvate. In this reaction 2 ATP molecule will be invest and 4 ATP molecule will be produced. 2 NADH molecule are also produced. Glycolysis consume 2 ATP and generate 4 ATPs. Thus, the process result in the generation of 2 net ATP. The process also generate 2 NADH. Glycolysis take place in the cytoplasm of every living cell. It is an important point to learn. Now, there are two phases of glycolysis. In the first phase, glucose is broken down to 2 molecules of glyceraldehyde 3-phosphate in a series of 5 reactions. In the second phase, another series of 5 reactions convert these 2 molecules of glyceraldehyde 3-phosphate into 2 molecules of pyruvate. Actually glycolysis total 10 steps पर मुश्तमिल एक प्रोसेस है. इसमें फर्स्ट 5 स्टेप्स को फर्स्ट फेज भी बोलते हैं और ये इन्वेस्टमेंट फेज भी इसको बोलते हैं. इसमें ये होता है कि ग्लूकोज के मॉलिक्यूल को ग्लिसरल्डिहाइड 3-फॉस्फेट के 2 मॉलिक्यूल में कन्वर्ट किया जाता है. जबकि सेकंड फेज भी 5 स्टेप्स पर मुश्तमिल है. इसमें 3 ग्लिसरल्डिहाइड 3-फॉस्फेट के जो 2 मॉलिक्यूल होते हैं उनको पायरुवेट के 2 मॉलिक्यूल में कन्वर्ट किया जाता है. Phase 1 consume 2 ATP and phase 2 generate 4 ATP. The net ATP production in the entire process is 2 reactions. Here there is the first reaction of glycolysis. In the first reaction, glucose molecule will be changed into glucose 6-phosphate. It is a phosphorylation of phosphate group. In this reaction, ATP molecule will be used. ATP breakdown into ADP, a phosphate group release. This phosphate group attached with glucose and glucose will be changed into glucose 6-phosphate. Hexokinase or glucokinase enzyme used in this reaction. This is one of the investment reaction. The phosphorylation of glucose takes place inside the cell. Hexokinase and glucokinase are isozymes. In reaction 2, glucose 6-phosphate will be changed into fructose 6-phosphate. Actually, it is a isomerization of glucose 6-phosphate. Glucose 6-phosphate convert into fructose 6-phosphate, and this reaction will be done with the help of phosphoglucose isomerase. Glucose 6-phosphate is an aldo sugar which will be changed into keto sugar, that is fructose 6-phosphate. In the third step or third reaction, fructose 6-phosphate will be changed or will be converted into fructose 1,6-bisphosphate. It is another phosphorylation reaction. In this reaction, ATP molecule break into ADP or phosphate group release. This phosphate will be attached fructose 6-phosphate, and this fructose 6-phosphate change into 1 fructose 1,6-bisphosphate. This reaction is done with the help of 6-phosphofructose 1 kinase, PFK1 or phosphofructokinase 1 enzyme involved in this reaction. In this phase, we invest our second ATP molecule. In step 4, fructose 1,6-bisphosphate will be changed. In steps 4, fructose 1,6-bisphosphate will change into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and this reaction is done with the help of aldolase enzyme. Glyceraldehyde 3-phosphate is used in glycolysis. Therefore, dihydroxyacetone phosphate has to be converted into glyceraldehyde 3-phosphate. This occurs in the next step. In reaction 5, dihydroxyacetone phosphate will be changed into glyceraldehyde 3-phosphate, and this reaction is done with the help of triose phosphate isomerase. This is the last step of phase 1, and at the end of this step there are 2 molecules of glyceraldehyde 3-phosphate. Student, in reaction 6, glyceraldehyde 3-phosphate will be changed into 1,3-bisphosphoglycerate, and this reaction will be done with the help of glyceraldehyde 3-phosphate dehydrogenase enzyme. The enzyme oxidizes the number 1 carbon aldehyde and then add a phosphate group. We have an acid anhydride in the product 1,3-bis phosphoglycerate. We have used an NAD for oxidation reaction. The cell has limited amount of NAD, so somewhere along the line we have to regenerate it, or glycolysis will stop. So in this step, NADH, a molecule of NADH is produced. In 7th step, 1,3-bis phosphoglycerate will be changed into 3-phosphoglycerate, and in this step ADP molecule, a phosphate group will be joined with ADP and 2 ATP molecule will be produced. In this step, this is the first step of energy production. We have recovered both ATP that were invested. Remember that each glucose gives 2 phosphoglycerate molecule. In reaction 8, 3-phosphoglycerate will be changed into 2-phosphoglycerate. Actually, it is a mutation, and this reaction is done with the help of phosphoglycerate mutase enzyme. phosphoglycerate mutase enzyme 3-phosphoglycerate को 2-phosphoglycerate में कन्वर्ट कर देगा. मतलब कि फॉस्फेट ग्रुप जो कि कार्बन नंबर 3 के साथ अटैच था उसको कार्बन नंबर 2 के साथ अटैच करवा देगा. In reaction 9, anolase enzyme will be used, and in this reaction 2-phosphoglycerate will be changed into phosphoenol pyruvate. Catalyze the dehydration of 2-phosphoglycerate to form phosphoenol pyruvate (PEP), and it is the last step of glycolysis. In this step, phosphoenol pyruvate will be changed into pyruvate, and in this step ATP also will be produced. ADP molecule, phosphate group attached with ADP and ATP produce. Catalyze the trans of phosphate from PEP to ADP to generate ATP and pyruvate. This is all about the glycolysis. Thank you. Please subscribe my channel Study with Umar. If you have any question, please ask me in the comments box.